So thanks a lot, and what I'm going to do is basically give you a very brief overview what we discussed over the day in terms of what could be important for the SGG oncology to focus on, what we should really concentrate, what we can do different, and also basically, of course, upcoming ideas for other projects we didn't have yet in scope or basically formalized. So I advance the slides here, I guess. So this has been basically the objectives, what we discussed throughout the day. So what do we want to focus on and how do we want to really transform cancer care? So what we discussed is basically that we want to move from a more static model, which cancer patients treated right now, meaning you identify at baseline the disease, you identify definitely mutations, or most likely in many cases you do, and then basically you treat the patient until the patient relapses. So we said we want to transform that in a more integrated, dynamic, personalized cancer care, which means we want to measure the mutational spectrum, the evolution of the disease, and also basically the consequence of the evolutionary pressure onto cancer more dynamically, basically following the cancer and the cancer evolution over time, making treatment decisions definitely more accurate at the moment they occur, and not only waiting until we see at a scan a new metastases or we basically see something growing or et cetera, really move it more into a direction that we follow the dynamic of cancer. As we all know, cancer is a dynamic disease. It's not static. It doesn't come up and then it stays the way it is. It really changes on many things. On the mutational spectrum, it changes on the microenvironment, it changes basically on the growth pattern, metastases, metabolomics, and that needs to be integrated if you want to make treatment decisions in the future in a little bit of a different way. That's why we called it basically Integrated Dynamic Personalized Cancer Care, or IDPCC. What it really entails is you can see here basically those 5 points. So we want to focus a little bit beyond patient stratification. I kind of mentioned it already, following the cancer patient over time, the evolution over time, making different treatment decisions. We want to also put it into context specificity, and or if you want to call it context dependency. That means a mutation depends on the context in which it occurs. So if you take a BRAF mutation, it's not only true that the patient has a BRAF mutation, he may have a P53 mutation, KRAS, NRAS, and so on. And the patient who has a P53 mutation, NRAS mutation, and BRAF is not the same as the one who is wild type on those genes. And that's something which we need to take much more into account when we make treatment decisions. And this is certainly one of the reasons why targeted therapy hasn't a response rate of 100%. It has a response rate from anything 40 to 70% at most, across the board. And that is basically certainly a reason why we need to take the context more into account. And we also need to understand the consequence of a mutation on a spectrum, like on the transcriptome, on the proteins, and on the metabolome. And that's kind of what we call basically multomics. That's a project where we try to get forward, where we look in the mutation and in the consequence of the mutation by really addressing what is the consequence of a mutation in the tumor on the proteome, on the transcriptome, and also basically on the metabolome. Those things needs to come together in a very integrated way to make different treatment decisions in the future. Second, of course, we need to talk about and we need to work on immuno-oncology, no question about it. This is the topic of the day, so to say, or of the decade. And there is also a project where we want to understand the tumor microenvironment much better than we do now. You all know that immune checkpoint inhibitors and targets, PD-1, PD-L1, and others, have a response rate 20 to 40%, depends on the setting. But those ones who derive a response, they really have a long-term response of years to come. And we want to really understand basically how can we select patients and how can we identify which patient should be treated with a checkpoint inhibitor and which patient may better be treated with a targeted therapy or a combination of those. And certainly we need to move away from the biopsies. We need to move away from a way that we want to punch into the patient every single time we see him. And this is also not feasible and even dangerous in the case of lung cancer and others. And in some cases, absolutely impossible. You have to think about GBM. And there is a tool basically which is called liquid biopsy. Liquid biopsy is ctDNA, cell-free DNA, RNA, and of course also other things, CTCs, exosomes, etc. So we want to look into projects basically which help us to address the diagnosis of cancer at baseline, but even more so longitudinally by looking into liquid biopsy instead of tissue-based diagnosis. And last but not least, also certainly a topic of the time now, is big data. Big data means how we can address and how we can capture what is collected in a daily community practice. If you think about how many patients are in clinical trials, it's just 3 to 5%. 3 to 5% of cancer patients participating in clinical trials, 95% don't. But the data generated on those 95% we are blinded to because they're not connected. They're sitting in institutions, they're sitting in payers, they're sitting in hospitals, they're sitting everywhere, but they are not connected. And we can learn a ton out of those data in terms of sequences, side effects, efficacy, and. And there we want to launch projects which are really looking into coordinating all of those big data, so to say, across Europe, and it's not going to be complete, but in really the major centers, that we can do cross-analysis and really understand what is the baseline of HER2-positive disease, what's the baseline of EGFR-mutated disease, etc., etc. And there are actually 3 projects on the way. One is called Harmony. Harmony is looking into big data in hematological disease. It's really focused on only hematological disease. This is a project which is close to be launched. And then we're looking basically in the replication of that in solid tumors. And one project of those we call this the Big 5 or the Big 3. We are not quite sure yet which kind of solid tumors we are taking. So that's a little bit up for debate. And another one is certainly focusing on prostate specifically because they were felt like it's a very specified topic that should be treated separately. So that's kind of the objectives. What have been the highlights of the day and what have we discussed? Certainly, access to innovative medicines have been very much a prominent topic of the day. How can we make innovative medicines compounds, number one, early accessible to patients, and number one, more widely accessible to patients? How can we make sure that every parts of Europe have the same access to the medicines and have the same, basically, possibilities to benefit from them? The other part was comparator arms. Do we really need to run clinical trials with comparators which have been trialed and tested many times? I'll give you an example. You take basically PD-L1 or PD-1 checkpoint inhibitors. There are, you name it, how many trials are done with PD-1, PD-L1 checkpoint inhibitors? Hundreds. Every single trial has a comparator arm. So why do we need to do that? Why do we need to continue to basically waste time, waste money, and waste actually patients' life by putting them on a comparator arm which is most likely inferior to something we know already? It's a regulatory problem, and that's also a problem, of course, in drug development, and that's something we also want to discuss and find ways to launch initiatives which may help to kind of generate a virtual comparator arm by maybe big data. We will see what the outcome is, but certainly not having 10, 15, 20, whatever trials using similar or even the same comparator arm. The other one was integrating of different endpoints into clinical trials, except of PFS, OS, side effects, and time to progression, clinical benefit rate, and what have you. More looking also PROs, patient-reported outcomes. How can we more work on that as a clinical trial endpoint? How can we more integrate imaging, standardizing imaging, which is still a problem. If you do an MRI on a Siemens and on a Philips machine, you not necessarily have the same data. And that's part of the discussions basically we had and we want to launch. Second is also basically, do we really need controlled clinical trials overall? If you look into the molecular profile, no 2 patients are alike. So what are we comparing it with? And that's something which we need to understand better and foster some research projects on that part, yeah? The other one is also outcomes for patients. So really, what is an outcome of a clinical trial which really benefits patients? And there, kind of what we said is this integrated dynamic personalized cancer care or precision medicine is really a tool to enhance and make outcomes better and make them different. And last but not least, every platform we are using should follow really the 3 criteria: achievable, affordable, and acceptable. And that's something which is really true, especially also looking for regulators, CDX, companion diagnostic, and those things across the world. What is able to register here in Europe is not necessarily what you can register in US. If you get a CE mark, you're not necessarily getting a PMA in the United States, and that's something which you definitely need to look into. So quickly, this has been the 4 projects, the 4 topics, essentially 5 projects Harmony, as I said, really focusing on the hematological part, mood omics, looking into the contextual dependency, what really a tumor entails in the overall holistic way, not only on a genetic spectrum. The Big Five and the prostate cancer initiative, so to say, as a replication of Harmony in solid tumors, and the tumor microenvironment to understand better who and when and how to put onto clinical trials. All in all, we discussed also what are the gaps and basically what is the so-called, what I call the gap check. So in order to make this live, to make this concept of integrated dynamic personalized cancer care really a reality and to bring it to life and make it sustainable, certainly everyone has to be on the table. Everyone has to work together in order to make it happen. So certainly we need to ensure patient access to innovative medicines, no doubt about it. We need regulators, EMA, FDA. It doesn't help if we only work with EMA or we work only with FDA. It's half of the truth only. We need patient advocacy organization, patient advocacy group. That's very, very important to involve patients early on and they can help us a ton in designing clinical trials, in designing projects, and really looking what constitutes outcome, what is something which is really meaningful and impactful. Certainly real-time evidence, what I said in terms of the big data, pricing models, we need to make sure the access is here. Safety is a very big point. I mean, as I said, certain safety aspects coming much, much later into play than we would see in clinical trials. Sometimes in clinical trials, you don't see much of side effects, but have a patient 2 years on a drug, you see something which you haven't seen in your clinical trial. That's something we really need to think. And last but not least, and nobody should take that personal, so silos are for farms but not for pharma. That means we really need to make sure that we collaborate. So we all bring those people and those patients and also those kind of interest groups, wherever they come from, and provide them a platform to really interact with us and work with us in order to make it really happen. And that's kind of the discussions we had over the day, focusing on those topics. With that, I thank you, and questions now or later?
Yeah, let's have it's time for a question or two right now. Any comments, questions? I have one.
Sure.
So you think that the industry is ready and mature enough for the industry to share data amongst themselves and with others? I mean, their own data?
Yeah. I mean, of course, I mean, this is a very big topic, as you know, data sharing in industry. I mean, there's no question about it. So I think there's ways to do that. I mean, for example, every trial we have done, every data which we generate on placebo arms, we can share because it's not competitive information. It's standard of care. So how a patient performs on a placebo arm is not a secret. So because it should be the same across industry. So those data we can really share and can make them available. If industry is ready completely to really kind of go into the experimental arm and make that available, I think that's a process. That's definitely a process, and mechanism needs to be discussed how that can be made happen. Yeah.